Welcome to Pharmacology Essentials, a concise micro-learning series designed for quick, practical understanding. In this course, we'll explore how drugs interact with the body, why they produce therapeutic or adverse effects, and how those insights inform safer, more effective treatments. Sit back and get ready for a clear, applied overview you can use right away. This short course is organized into 3 clear chapters. Core concepts that build foundational knowledge, drug action which explains how medicines work at the molecular and cellular level, and dose and response covering how dosage shapes therapeutic effects and safety. We'll move through each section with practical examples and visual summaries to help you retain the essentials. Now we begin Chapter 1, Core Concepts, where we set the stage for everything that follows. In this chapter, we'll define pharmacology, outline its scope, and highlight how it draws from chemistry, physiology, and pathology to turn molecules into medicines. Think of this as the conceptual toolbox you'll use in later chapters. Pharmacology is the study of drug action on living systems, combining chemistry, physiology, and pathology to predict effects and design therapies. It covers drug discovery and mechanisms, and aims to maximize benefit while minimizing harm. The visuals here emphasize the cross-disciplinary nature of the field and why understanding basic principles matters for clinical decisions. Pharmacokinetics looks at what the body does to a drug, summarized by ADME: absorption, distribution, metabolism, and excretion. These processes shape concentration time profiles, which determine onset, duration, and intensity of effect. When you view pharmacokinetic curves, you can predict how dosing and route influence therapeutic levels and potential toxicity. Chapter 2 focuses on drug action and dives into how drugs interact with targets to change physiology. We'll explore receptor theory, different receptor families, and how molecular interactions translate into whole organism responses. This chapter connects molecular binding events to the clinical effects clinicians observe and manage. Receptor theory explains how drugs produce effects by binding specific proteins on or inside cells. Key ideas include the dose-response relationship, receptor occupancy, and the variety of receptor types like G-protein-coupled receptors, ion channels, and nuclear receptors. These concepts help predict how changing concentration or affinity alters the magnitude of response. Agonists activate receptors to produce a biological response, while antagonists prevent activation and block that response. Partial agonists provide intermediate activation, and inverse agonists can reduce baseline activity. Understanding these types helps explain drug behavior in different clinical contexts and supports rational treatment choices when multiple agents are available. We now move into Chapter 3, Dose and Response, where we analyze how varying doses change effects and safety. This chapter ties together pharmacodynamics and pharmacokinetics to explain why dose selection matters, how we compare drugs, and what guides therapeutic decisions in real-world practice. Graded dose-response curves are typically sigmoidal, showing how response increases with concentration until a maximum effect. 2 key properties are efficacy, the maximal effect a drug can produce, and potency, the concentration needed to achieve a given response. These curves guide therapeutic dosing and help compare drug candidates during development. Quantile or population dose-response curves describe how individuals vary in their minimal effective or toxic doses, letting us estimate ED50, TD50, and LD50. The therapeutic index, the safety margin between effective and toxic doses, emerges from these data. Population variability from age, genetics, or comorbidities underlies the need for personalized dosing strategies. Routes of administration and biological barriers critically shape drug delivery. Oral, intravenous, intramuscular, sublingual, transdermal, and inhalational routes differ in onset and bioavailability, while membrane properties like lipophilicity and transporters influence absorption. Patient acceptability, convenience, pain, and speed also affects route selection in clinical practice. First-pass metabolism occurs mainly in the liver after oral dosing, substantially reducing bioavailability for some drugs. This effect means oral doses may need to be higher or alternative routes chosen to achieve therapeutic levels. The photo here reminds us that practical considerations like formulation and route determine real-world drug exposure. Drug metabolism involves phase 1 reactions such as oxidation, reduction, and hydrolysis that introduce reactive groups, and phase 2 reactions like glucuronidation or sulfation that increase water solubility. Together with specific metabolic pathways, these steps determine elimination and potential interactions, so understanding them helps predict dosing and drug-drug effects. Renal clearance removes many drugs through glomerular filtration, tubular secretion, and reabsorption, and kidney function directly impacts drug elimination. In patients with impaired renal function, dose adjustments are often necessary to avoid accumulation and toxicity. Monitoring renal function and adapting regimens is a core safety practice in pharmacotherapy. Drug interactions can alter therapeutic outcomes through multiple mechanisms, including enzyme induction that speeds metabolism, enzyme inhibition that slows it, displacement from plasma proteins that increases free drug levels, and receptor-level antagonism that blocks effects. Recognizing these interactions is essential when managing polypharmacy and preventing adverse events. Adverse drug reactions range from mild side effects to severe, life-threatening toxicity. Early recognition, pharmacovigilance, and sometimes genetic screening improve patient safety and guide safer drug development. Staying alert to unexpected signs and reporting suspected reactions helps protect patients and refine treatment recommendations. Thank you for joining this Pharmacology Essentials overview. We've covered core concepts, drug action, and dose-response principles to help you make smarter, safer decisions with medicines. Review the visuals and key points as needed, and feel free to revisit any chapter to reinforce your understanding before applying these ideas in practice.